Assalamualaikum and hi everyone. Thank you for joining us today. My name is Nurul Akila and I am delighted to be here to share our presentation on mutation breeding by chemical method. Before we dive into the details, I would like to introduce you to my partner Ifano Widat binti Rafidi and me, Nurul Akila binti Ramli.
So, firstly, we need to know the list of definition. So, what is mutation? Mutation is when there is an accidental and heritable changes in the character of a plant. There are two types of mutation, which are spontaneous mutation, which is in nature spontaneous mutation occur, and the second one is induced mutation, which is mutation can be induced by treating cells with mutagens during the S phase of the cell cycle. Mutagenesis is a process of inducing mutations. Mutagens is the physical and chemical agents which greatly enhance mutations. So there are two types of mutagens. The first one is physical mutagens and the second one is chemical mutagens. So lastly, we need to know what is the meaning of mutation breeding. So mutation breeding is the utilization of induced mutation for crop improvement. So the first one is alkylating agents. It transfers the alkyl group to DNA. This transfer occurs at the end ring of guanine. For the examples, AMS, MMS, AAS, and the last one is AI. So this is the mode of action. For AMS, AT to GC or GC to AT transitions, MMS transitions, AAS, GC to AT or AT to GC transitions, and the last one is AI, which is transitions. The second one is base analogs. The molecules of these chemicals are very similar in structure to DNA bases. Thus, they are easily incorporated into DNA during DNA replication. For the examples, bromouracil and aminoporine. The mode of action of bromouracil and aminoporine is AT to GC or GC to AT transitions.
Now that we have established the groundwork, it's time to explore a key aspect that sheds light on dose of mutagen and its impact on mutation breeding by chemical method. The mutation treatment reduces germination, growth rate, and fertility of organism. Damage increases with increase in the dose mutagen and duration of exposure. Optimum dose of mutagen is expressed as LD50. LD50 refers to a dose of mutagen that kills 50% of the treated individuals. The dose of treatment depends on the intensity of radiations or concentrations of chemical mutagens. An optimum dose is the one which produces the maximum frequency of mutation and causes minimum killing. Dose close to LD50 should be optimum.
Next is targeting induced localizations in genomes, which is TILING. TILING is a powerful reverse genetic strategy that employs a mismatch specific endonuclease, which is an enzyme that identifies a mismatch and cuts the double-stranded DNA at the specific site. The function is to detect induced or natural DNA polymorphisms. It also allows the rapid and cost-effective detection of induced point mutations in populations of chemically mutagenized individuals. So this is the TILING technique process. Next is giving mutagens treatment. Firstly, X-ray irradiation is given in X-ray room. Gamma ray irradiation is given in gamma garden. Next, neutron treatment is given from neutron flash coming from generator. And lastly, beta rays treatment is given in the radioisotope laboratories.
The next step using chemical method is irradiation of plant breeding. Mutant selection is the process of identifying plants that have been improved by induced mutation through irradiation. The first step is M0. M0 involves mutagenesis of seeds, pollen, vegetative parts or tissue cultures. Second, M1. Plants grown from treated seeds, M1 or vegetative propagule, M1V1. Third step is M2. M2V2. Population of plants grow from seeds M2 or vegetative parts M2V2. Selection of desired mutants may start in this generation or later. The next step is M3 to M2. Continued selection, genetic confirmation, multiplication, and stabilization of field performance of mutant lines. Next, 2-3 generation. Comparative analysis of mutant lines during different years in different locations. The last step is next 2 to 3 generations. It is official testing before release as new variety. The last chemical method that we use in plant breeding is phage mutagenesis, method that involves the use of cosmic rays in outer space to induce spontaneous mutations. Moreover, cosmic rays and microgravity affect plant biology itself. This is being explored in astrobotany studies for a different purpose. These are the steps involving in phage mutagenesis. First, plant seeds are taken to outer space. Second, radiation from cosmic rays is used to speed up the evolutionary process in plants. Third, once returned to Earth, the seeds are germinated in laboratories and observed to see whether new useful traits have been induced. The last step is scientists analyze whether the mutation induced in the harsh conditions of space can help astrobotany and plant breeding on Earth, where plants endure harsh conditions imposed by climate change. In conclusion, it's important to note that while plant mutation breeding can be a valuable tool for a crop improvement, careful evaluation and regulatory considerations are necessary to ensure the safety and potential benefits of the mutated plants for human consumption and the environment.
That's all from us. Thank you so much for listening to our presentation.